Hello everyone. In this session, we will study class 9 chapter tissues. So let's see what all topics we are going to cover in this chapter. First, we'll see what are tissues and why are they needed. Then we will compare plant and animal tissues. Then in this session, we will focus mainly on plant tissues and their types: meristematic, permanent and protective tissues one by one. So let's begin. As we all know, all living organisms are made up of cells, and on the basis of number of cells, we can categorize living organisms into unicellular and multicellular. As the term suggests, uni means one. Organisms that are made up of only one cell are called unicellular organisms, and organisms that are made up of multiple cell or millions of cells are called multicellular organisms. Let's see the examples of unicellular organisms first. All bacteria, E. coli, Streptococcus, Halobacteria are unicellular. Then all protists, amoeba, euglena, basically protozoans, paramecium are unicellular. Then some of the algae like chlorella, chlamydomonas are unicellular. And then yeast is also a unicellular fungus. Means all these organisms are made up of only one cell. Now bacteria are, as we know, they are prokaryotic. Algae, fungi, we know are eukaryotes. So unicellular organism, as you can see, can be prokaryotic or eukaryotic. Now let's see the key difference of unicellular from multicellular apart from the number of cells. Unicellular organisms, the one cell that is present in them is not specialized, means it does not have a specific shape or size or a function. It can do all the functions that are needed to support the life of that organism. Let's understand it with the help of amoeba. Amoeba is a unicellular organism, and this single cell is capable of doing all the functions that are needed to support the life of amoeba. For example, this cell membrane here can perform the function of gaseous exchange. So it can perform the function of respiration. This cell membrane is capable of waste removal also. So we can say it can perform excretion. This cell membrane, along with the food vacuole that is present, is capable of performing the function of ingestion and digestion. Pseudopodia also helps in locomotion, movement. And the nucleus that is present in amoeba is capable of undergoing binary fission. It can perform the function of reproduction. So a single cell is capable of doing all the life processes that is necessary to support the life of this organism. Now let's see the case of multicellular organisms. Us human beings, all plants, animals, birds, insects, amphibians, reptiles, all fungi, all algae are multicellular organisms. Multicellular organisms are made up of millions of cells, but they have specialized systems. Different cells are specialized to perform a specific function. For example, let's see the case of animals. In humans, muscle cells are there to perform the function of movement. Nerve cells are there for the transmission of messages, or you can say responding to the external environment. Then blood is there for the transport of oxygen, nutrients, hormones, waste, etc. So different cells are performing different functions. So there is distribution of work which makes them more efficient than unicellular. Now let's see the case of plants also. Different group of cells, we call them xylem, are performing the function of transport of water and nutrients. Another group of cell, we call it phloem, is performing the function of transport of food, and there are many other such group of cells that are working together to perform different functions. Now, this distribution of work, or you can say division of labor, makes these multicellular organisms more efficient than unicellular one. They can perform that one particular function more efficiently because of it, and that also increases the lifespan of these multicellular organisms. Now let's go a step further and understand the level of organization in case of multicellular organisms. Different cells group together to perform a specific function, and this group of cell we call it tissues. Different tissues work together for a specific function, we call them organ, like heart, pancreas, liver, etc. Different organs are working together to form an organ system like digestive system, circulatory system, excretory system, etc. Different organ systems work together to make an organism function properly. Similar organization we see in case of plants also. Like different cells are working together for performing a specific function. They group together to perform that particular function, and we call this group of cells as tissues, like dermal tissues, providing protection, vascular tissue for conduction, and ground tissues for storage of food, performing photosynthesis, etc. So these tissues, these are present in case of plants. They group together to form an organ like leaf, stem, root, and different organs work together to form organ system like shoot system, root system, and all these organ system work together to make plant function as an efficient organism. So now let's focus on our main topic, which is tissues. What are tissues? Tissues are group of cells similar in structure because of their common origin, and they work together to perform a specific function. For example, blood and muscle are examples of tissue in case of animals. Phloem, xylem are example of tissues in case of plants. First question that comes to mind is plants and animals both have tissues, and are these tissues similar? Are plants, animals made of same type of tissues? So let's see. Animals generally have these four main types of tissues: connective tissues, epithelial tissue, muscle tissue, and nervous tissue. Connective tissue, as the name suggests, help in the connection of different tissues, other tissues and organs within the body. Epithelial tissue provides protection from the external environment, and also provides kind of isolation to different organs. Different organs are covered with epithelial tissue, so it kind of keeps them isolated from each other so that they can perform their function. Muscle tissue helps in the movement, also helps us in standing upright, pushes down the food also. Nervous tissue helps in transmission of messages, for responding to the external environment. So these are the tissues that we see in case of animals: connective, epithelial, muscle, nervous tissue. Let's see the case of plants. Plants have all these tissues, and we're going to see them in detail in our coming slides. But they have vascular tissues like xylem and phloem for the conduction of water, mineral, and food. Xylem, phloem, group of cells forming tissues, and they are helping in the conduction of water and food. Ground tissues help in basically storage of food, photosynthesis, and many other function forms. The body primary growth of the plant happens with the help of these tissues. And then we have dermal tissues, epidermis, etc. They basically provide protection, right? So we can say plants and animals have different kinds of tissues in them, but why is it the case? Let's understand the difference between plants and animal to get the answer. So plants are stationary. They have a sedentary lifestyle. They do not move. So they need more of mechanical tissue, supportive tissues to stand upright. And these tissues need not be alive. They can be dead. So they mostly contain dead cells. Whereas if we see the case of animals, they are capable of movement. They show locomotion. They move for obtaining food, for shelter, for mating, for finding their partners. So they need energy for all these activities, like searching for food, shelter, mates. And for energy, they need to perform metabolic activities, or they need to show metabolism. So the tissues that are present in case of animals are living tissues mostly. Now let's see other differences. In case of plants, growth is restricted to certain regions. Growth is not uniform. As the plant develops, all plant parts are not developing at the same rate. This is because they have a division of dividing and non-dividing tissues. They have these two type of tissue, and dividing tissues are localized. They are present in certain regions, so that makes the growth of plants non-uniform. Now let's see the case of animals. They do not have clear separation of dividing and non-dividing regions, and they grow uniformly because of it. Organ systems are specialized and localized. So as you can see, plants and animals have different structure, different functions, or you can say they have different modes of life. One is sedentary, other show a lot of locomotion. They have different feeding habits as well. So because of the difference in the structure and because of the difference in the type of functions that these tissues need to perform, their structure is also very different. Because of the function that plant needs to do, it needs to have different tissues from the animals. First, we will study plant tissues in detail. So let's start with plant tissues. Plant tissues, on the basis of kind of cells that they contain, can be categorized into meristematic, or you can say dividing tissues, and permanent. As the name suggests, they are permanent. They do not divide. So non-dividing tissues. So plants have mainly these two type of tissues, and can be categorized further into subtypes. And in the coming slides, we will study all these types of tissues in detail one by one. So let's begin with meristematic tissue. Meristematic tissues are dividing tissues. They are undifferentiated. They are not specialized. They are a group of undifferentiated cells that are capable of undergoing division, continuous division. So if we see the structure of meristematic tissue, it looks something like this under the microscopes. These are group of cells. And if we see the structure of one cell, it has a very thin cell wall. A very prominent nucleus is present in the center. The cytoplasm in the cell is very dense. So dense cytoplasm, prominent nucleus, and thin cell wall. And you can see the intercellular spaces between two meristematic cells is not there. There are no intercellular spaces. Vacuoles, chloroplast are also absent. So these are also absent. So this is how meristematic tissue looks like. And one key property of meristematic cell is that they are capable of undergoing division continuously and forming cell exactly like themselves. We call this property as pluripotency. So these cells can divide and form other cells exactly like themselves. And both of these cells can divide even further. So they are pluripotent cells. So meristematic tissues are made up of actively dividing cells that are responsible for the primary growth of the plant. Then cells in these tissues are undifferentiated. They do not have any specific shape, size, or function. They are not specialized. Meristematic cells are pluripotent. They can develop into different cell types. Cells are small, thin-walled, densely packed with dense cytoplasm. Then last, vacuoles and chloroplasts are absent as the focus of the cells is basically on division. not storage or photosynthesis. So these are meristematic tissue. Now let's see where are they located in case of plants. They are located only at specific regions in plant, and because of their specific localization in the plant, they give a special pattern to the growth of plants. Let's understand it with the example of animals. This is the slide showing development of a adult male. As you can see, arms, legs and torso grow uniformly at the same rate as the man develops. So growth is uniform. All the parts, arms, legs, torso, are growing at the same rate as the man develops. Now, if we look at plants, and this is the slide showing growth in plants. If we see growth in case of plants, root and stem show more growth compared to other parts of the plant. So growth is not uniform. As the plant grows, some parts show more growth compared to other parts. They are not growing at the same rate. And this is because of the localization of meristematic tissue. Meristematic tissues are localized in plants. Some are present in the apex, shoot apex, root apex. In case of monocots, we see intercalary meristematic tissue. So they are located at specific regions in the plant. So the growth is seen only in those regions of the plant. That makes the growth in case of plants non-uniform, right? On the basis of localization of these tissues, meristematic tissues can be divided into three types: apical meristem, intercalary meristem, and lateral meristem. Apical meristem basically present at the apices or tips, like shoot apex, root apex. Intercalary meristem we see in case of monocots, grasses. This is the stem of the plant. This is internode. This is one node, node 1, and this is the second node. Intercalary meristem is present at the base of internode and at the base of leaf. So intercalary meristem is present at the base of the internode and the leaf base. And lateral meristem, as the name suggests, is present laterally in the stem and roots. So let's see these three type of meristematic tissues one by one. Apical meristem. Apical meristem is present at the apex of the shoot. We call it shoot apical meristem or SAM. Root apical meristem. If we cut the tip of the root, see it under the microscope, we will see something like this. This is the root cap that is basically protecting the root from damage in the soil. Now this is the root apex, and meristematic tissue is present in this region. We call it root apical meristem. So apical meristem can be of two types: shoot apical meristem and root apical meristem. And these two kinds of apical meristem are responsible for increasing the length of root and shoot. Shoot apical meristem will help in increasing the length of stem, and root apical meristem will help in increasing the root of the plant. So we can say they help in the primary growth of the plant. They help in increasing the length of the plant. Apical meristem. Now let's see lateral meristem. Now this is a figure showing plant, and if we cut the stem transversely, we take a cross section of the stem, we are going to see something like this under the microscope, right? So these are the vascular bundles, xylem and phloem. and they have a layer of meristematic tissue in between them. Also, between these two vascular bundles, this layer here is made up of meristematic tissues. So it forms a kind of ring, all the meristematic tissue between the vascular bundle and inside the vascular bundle here. It forms a ring, and we call this ring as vascular cambium. This is one type of lateral meristem. It is capable of cutting off cells inside as well as outside. This vascular cambium can cut off cell inside and as well as outside. When it will form more cell, it will help in increasing the thickness of the plant, or you can say girth of the plant. So this is lateral meristem. It helps in increasing the girth in the plant. Second type of lateral meristem that we see in case of older stem, it is cork cambium. First one was vascular cambium. Second is that we see in case of older stem, cork cambium. Some of the cells in the cortical region, cortex, will become meristematic. Some cells here, like in cortical region, will become meristematic, and they will cut cells on the inner side as well as on the outer side. They will divide and form new cells on the inner side also, on the outer side also. And it will also help in increasing the thickness of the plant. In fact, it cuts off so many cells that it crushes the outer epidermis. Epidermis is gone and only cork cells are visible in older stems. So these are the two types of lateral meristem that are present laterally in the vascular tissue and in the cortical region. Now, third one is intercalary meristem that we see in case of monocots, grasses, like sugarcane, bamboo, mint plant, etc. So this is a sugarcane stem, you can see. This is one node. This is another node. If I ask you the definition of stem, it will be like it has nodes and internodes. So one node, second node, and between these two nodes, we have an internode. So intercalary meristem is present at the base of internode, or you can say it as present at the leaf base also. So it helps in increasing the length of an internode in case of monocots, in case of grasses, right? So these are the three type of meristems on the basis of location. And they are, because of their presence at only specific regions in the plant, plants only show non-uniform growth. They show growth of certain parts more than the other parts. This gives a very unique pattern of growth to plants. Now let's see the functions of meristematic tissues. Meristematic tissue enables growth in plant in length, girth, and also helps in regeneration. So basically we can say they help in the primary growth as well as secondary growth in plants, meristematic tissue. Second is they produce new cells that later become specialized. So if we see a meristematic cell, it undergoes division and forms new cell. After some time, these new cells will also undergo division. But after a few divisions, some of these cells loses the capability of division. They attain a specific shape, size, and function. Depending on where they are located in the plant, they will attain the particular shape, size, and they become specialized, you can say. Or we can say differentiated. So, meristematic cells give rise to specialized cells later on, right? Third function of meristematic tissue, they help in wound healing. Whenever some kind of damage happens to plants, these cells, some of the cells, meristematic cells, will help in will form new cells and help in the repair because of the wear and tear that happens to plants, right? Now, let's start with second type of tissues, which are permanent tissues. So, permanent tissues are group of cells that are specialized. Means they have specific shape, size, and functions. They are permanent. They are not able to undergo divisions. They perform a specific function. They are differentiated. How are these permanent cells formed in the body? Permanent cells are formed from meristematic tissues only, from meristematic cells only. Meristematic cells undergo division and form cells exactly like itself. Means those cells are also capable of division initially, but after some time they lose the capability of division, and on the basis of their location in the plant they attain a specific shape, size, and function. Then we call this cell as specialized cell or a differentiated cell. Means, for example, if it's located in the leaf, this cell will form a for example, it might form a mesophyll cell. In case of phloem, it might become a sieve cell. And in case of root, it might take the structure of a epidermal cell, will form a root hair, as you can see. So on the basis of their location, these meristematic cells, eventually with time, they become permanent. They become specialized. They take a specific different kind of shapes, and they become specialized to perform definite function, right? Specific function. So permanent cells are produced by meristems that are very similar to meristematic cells. As these cells grow and mature, they differentiate or they become specialized. So this process of formation of permanent cell from meristematic, we call this process as differentiation. It leads to formation of permanent tissue from meristematic. Means permanent tissue, that cells that have specific structure and function. Now let's see the categories, subtypes of permanent tissue. We can categorize permanent tissue on the basis of type of cells that it contains. For example, it can be categorized into simple and complex permanent tissue. Simple permanent tissue, as the name says, it's very simple. Same type of cells grouped together, same type of cell grouped together, same structure, performing the same function, they group together, those permanent cell grouped together to perform a function, we call it simple permanent tissue. Complex permanent tissue, different types of cells which are different in structure. So cells, they group together to perform one common function. We call it a complex permanent tissue in this case. So let's study simple permanent tissue first. Simple permanent tissue is usually found below the epidermis. If we cut the section of stem or root, first layer would be epidermis. Below this epidermal layer, epidermis, we generally see simple permanent tissue. These are the cortical cells. These are the simple permanent tissue. And they are made up of only one type of cell, simple permanent tissue. On the basis of structure and function of cells, we can categorize simple permanent tissue into three types: parenchyma, collenchyma, and sclerenchyma. The name is little sclerenchyma. Little mistake here, sclerenchyma. So let's study these three types of simple permanent tissue one by one. First is parenchyma. Parenchyma are most common simple permanent tissue. They are present in stem, root, leaf, flower, seeds. You see parenchyma tissue everywhere in the plant. Now, if we see simple parenchyma cells and we cut the transverse section, like this is the stem and we cut a transverse section, the parenchymatous cells look something like this: round, oval, polygonal cells. So you see these cells in transverse section, something like this. And if we cut the longitudinal section along the length of the plant, these cells appear elongated. Let's see some of the properties of parenchyma cells. These cells have thin flexible cell wall, just like meristematic cells, thin cell wall. Prominent nucleus is present. Prominent nucleus is present. Then these cells have vacuoles in them, which was not in meristematic cells. They have large vacuoles present in the center. These cells have intercellular spaces between the cells. These are simple parenchymatous cells, and you see intercellular spaces between the cells. They also have dense cytoplasm in them. So if we see these three properties, thin cell wall, prominent nucleus, dense cytoplasm, it is very similar to meristematic cells. But they differ from meristematic cell because they have large vacuoles. Vacuoles are present, and then they have intercellular spaces between the cells. So these are the simple parenchymatous cells. And what are the function of simple parenchymatous cells? Parenchymatous cells, their main function is storage of food. They are present in stem, root, leaves, seeds, etc. And the major function that they perform is storage of food. Carrots, which we know are roots, they store food in their parenchymatous tissues. Potatoes, underground stems, and they store starch in starch granules in parenchymatous. These granules are present in parenchyma cells. And then coconut, which are basically seeds, store food. This is all the pulpy material in fruit is made up of parenchyma, and they store food. Pulpy material that you see in fruit are basically they have parenchymatous tissue, and the function is storage of food. The next function that they perform is photosynthesis. If we see, this is leaf of the plant, and if we cut a cross section, it will look something like this under the microscope. This is the cuticle, uppermost layer, which is basically waxy layer secreted by epidermal cells. This is the upper epidermis. And these tightly packed layers, these tightly packed layers here, these are basically palisade parenchyma cells made up of parenchymatous tissue. These are parenchyma cells. They have chloroplast in them. This is the vacuole, and these are green colored structure are chloroplast. These cells perform the function of photosynthesis. They receive the light, they have chlorophyll in their chloroplast, and they perform the function of photosynthesis. We call this parenchyma chlorenchyma. A special name has been given, chlorenchyma. These parenchyma are called chlorenchyma. Then we have spongy parenchyma, and their function is storage of food. And then we have lower epidermis. So second function is photosynthesis, and we call this parenchyma as chlorenchyma. So the third function that we see for parenchyma cell is in case of aquatic plants. Parenchyma cells have been given special name in case of aquatic plant. It is called aerenchyma. Air. Air cavities are present between parenchyma cells, many air cavities. So if we see example of aquatic plant, this is lotus, and if we cut a section, transverse section, that section is going to look something like this under the microscope. So if we enlarge this, these are parenchyma cells, and they have air cavities. We call these air cavities as lacunae. Empty spaces present, these lacunae are present between parenchyma cells in case of petiole of lotus. Why is it the case? Why air cavities are formed? These air cavities helped in two things. One, it helps in gaseous exchange. These are aquatic plants. So oxygen and CO2, which is basically in water, it is lesser. So for easier exchange of gases, air cavities are present. They can easily flow through these, air can easily flow through these cavities. Another function is these cavity makes the petiole lighter, so it provides buoyancy to the plant. It helps, this lighter weight will help the plant in floating easily. Let's also understand how parenchyma is formed, how these air cavities develop in the petiole. Now, if we see this portion, initially there are only parenchyma cells. There is no air cavity. But after some time, during growth, some of the cell undergo programmed cell death, some of the cell die by apoptosis, and air cavities develop between parenchyma cells. So this lacunae, air cavities develop, and they provide buoyancy and help in the gaseous exchange. This is the example of another aquatic plant, water hyacinth. And if we see the stem of this plant, we see air cavities. This is air cavity, and it is lined by parenchyma cells. And they have a particular diameter also, a range of diameter also of these air cavities. So parenchyma tissue is present in the water hyacinth and many other aquatic plant, locust, lotus, ecornia, etc. Now let's see second type of simple permanent tissue, which are collenchyma. Collenchyma cells are also living, just like parenchyma cells. These are living cells. And if we see these cells in the transverse section, they appear something like this. So these are polygonal cells with very little intercellular space between them. A special feature of collenchyma cells is that they have thickenings at the corners in their cell wall. So this is the cell wall. Thickenings are present only at the corners, not along the entire cell wall. It is only present at the corner. They have very little intercellular spaces between them. They have nucleus, a large vacuole is present, and the cell walls have thickening only at the corners. Now, if we see the longitudinal section, collenchyma cells appear very elongated, and they have thickenings at the corner. Very little intercellular space between them. They have chloroplast, nucleus, large vacuole is one of the key features, and cytoplasm in them. So these are the collenchyma cells. As you can see, these cells have thickenings at the corners. So they must be present in plant parts that need support, but not too much rigidity, because the thickenings are not present along the entire length of cell wall. It provides flexibility also. So wherever moderate amount of strength along with flexibility is required, collenchymatous tissue is seen in the plant part. For example, if we see their location, they are found in the leaf petiole, here leaf stalk. This leaf needs to move to receive maximum amount of sunlight. So this collenchymatous tissue, which is present just below the epidermis, provide strength, mechanical strength along with flexibility. Collenchyma cells are also present in the veins around vascular tissues. It is also present in tendrils and climbers. Since they need to wound around another stem for support, flexibility is needed along with moderate amount of flexibility along with moderate amount of strength is needed. So it provides, collenchymatous tissue provides both. So these are the parts where collenchymatous tissues are located. Now, if we see the cross section of vein of leaf, we are going to see something like this under the microscope. This is the upper epidermis. This is palisade parenchyma. Then you have spongy tissues, lower epidermis. And here, just below the epidermis, in the vein region, we see collenchyma cells. Right? So, wherever moderate amount of strength along with flexibility is needed, collenchymatous tissue is present. So the third type of simple permanent tissue are sclerenchymatous, sclerenchyma tissues. These are made up of dead cells. Collenchyma and parenchyma are living cells, but sclerenchyma are dead cells. If we see these cells under the microscope, in the transverse section, these cells look something like this. No intercellular spaces, and cell walls are lignified and thick. And if we see these cells in longitudinal section, they are long, they are elongated. And if we see these cells in longitudinal section, they look elongated, very long, and they have thickenings on their cell walls, making the lumen very narrow. Lumen is very narrow. So these are long cells, elongated cells, narrow lumen, and cell walls are thickened. Cell walls have thickenings. They are lignified. They are thick and cell walls are lignified. There is no intercellular spaces between mature cells. Now, their main function is they make the plant part rigid and stiff. They provide rigidity and stiffness to the plant. So let's see, where are they located? They are found in the stems, veins of the leaves, seed coats, nuts, and coconut husk. So if we see the cross section of the stem and leaf, around the vascular tissue, we see sclerenchymatous tissue. So this is a section of stem, this is xylem, phloem, vascular bundles, and in these regions, we see around the vascular tissues, we see sclerenchyma. So this is the section of a leaf, in vein region, where vascular bundles are present, we see sclerenchymatous tissue. In the coconut husk also, we find these sclerenchyma cells. These are dead cells. The main function is they provide strength and rigidity to the plant part. So now let's study complex permanent tissue. Complex, as the name suggests, there are more than one type of cells that group together to perform one function, one common function. Different cells work together to perform a common function. Now, on the basis of function, we can categorize complex permanent tissue into xylem and phloem. Phloem and xylem, they are basically responsible for the transport. Xylem does the transport of water and minerals from roots to the aerial parts of the plant. Unidirectional transport from the roots to aerial, to stem, leaves, to all aerial parts of the plant. And phloem is responsible for the transport of food, amino acid, hormones, organic acids, etc. from source to sink. Let's understand source and sink here. Source is part which has abundant food available, like leaves, which are manufacturing and preparing the food. So from leaves, it is generally transported to the parts where food is required. For example, roots and the lower leaves, if they need food, since younger leaves and the leaves at the top are able to manufacture more feed, or to any storage organs like underground stems, etc., wherever food availability is there, food requirement is there. Sometimes from the stored organ like underground roots or stem, food is transported to leaves. So transport of food is always from the place, from the plant part where food is available in abundance to the part where food is needed or required. So we can say it is a bi-directional transport. It is a bi-directional transport from source to sink. And generally it is mostly from leaves to the stem and root and underground parts. So xylem and phloem, are the conducting tissue that are responsible for the transport of food, water and minerals in the plants, and together they form a vascular bundle. Xylem and phloems are generally arranged in the form of vascular bundles in plants. Vascular tissue helps plants survive on land by transporting materials. So these two are basically responsible for the transport in plants. Now, one more thing that you should know is that true land plants, first terrestrial plants are pteridophytes and they are called true land plants, and bryophytes are not considered true terrestrial plants. Rather, pteridophytes are considered true terrestrial plants, even though bryophyte exists on soil only. Pteridophytes are considered first true land plants because they have xylem and phloem conducting tissues in them. So these are the complex permanent tissue. Now let's discuss xylem and phloem one by one. Xylem has four types of cell: tracheids, vessels, xylem parenchyma, and xylem fibers. These three types of cell, tracheid, vessels, and xylem fibers, these are dead cells. These are dead cells. Only living cell type in case of xylem is xylem parenchyma. So let's understand the structure of these four types of cells in xylem. So tracheids are elongated tubular structure with tapering ends. Tapering ends. Their diameter is lesser compared to the vessels. They are longer than vessels, but their diameter is smaller compared to the vessels. So they are elongated structure with tapering ends. They have pits on their side walls. So if two tracheids are arranged like this, if two tracheids are arranged side by side like this, two tracheids are arranged like this, then from this particular tracheid pit that is present in the side wall of this tracheid, water can travel to another tracheid through the pit of that, from pit of one tracheid to the pit of another tracheid, like this. Water can travel like this also, right? So through pits water can move. So there are tracheids have pits on their side walls. Now coming to the second member, vessel. Tracheids are dead cells. Similarly, vessels are also dead cells. They do not have nucleus, cytoplasm, organelles. They are dead cells and their walls are lignified. Lignification in the cell wall is there. These are vessels. These are smaller, shorter compared to tracheids, but have larger diameter compared to tracheids. And their end wall, they are arranged end to end. Like vessels are arranged end to end, and their end walls are perforated. So water can easily move from root to leaves. There is unidirectional transport of water and minerals from roots to leaves via vessel. These are the main conducting cells. Tracheids also conducts water and minerals, but main conduction happens via vessels present in angiosperms. Third is xylem parenchyma, and their main function is storage of food, right? And fourth member, xylem fibers. These are also dead cell elongated cells and their walls are lignified. Lumen is narrow lumen. It is very narrow and it basically provides support. So these four types of cell group together and they allow the vertical transport of water and minerals from the roots to the leaves. Main function of xylem is transport of water and minerals from roots to other aerial parts of the plant. Now let's discuss the second complex permanent tissue, that is phloem. Phloem also has four components: sieve tube, we can call it sieve tube element, then companion cell, phloem parenchyma and phloem fibers. All okay. In sieve tubes are present in modern land plants, that is most of the angiosperms. But one thing you should know is, in some gymnosperms and primitive angiosperms, we see structure that is primitive to sieve tube, sieve cells. Instead of sieve tube element, sieve cells are present in some of the gymnosperm plus some primitive angiosperms. Now, sieve tube element or sieve cell, companion cell, and phloem parenchyma all are living cells. Only dead component in phloem is phloem fibers. Phloem fibers are dead cells. All these four type of cells group together to form phloem, food conducting tissue in plants. Let's see the structure of these components. So sieve tube element is a tubular structure. It's an elongated cell, as you can see here, with perforated end walls. And they are also these perforated end walls, and they are also connected end to end. The cytoplasm is reduced and form a thin layer at the side. And very few organelles are present, very few mitochondria. These cells are living with the help of companion cells which are present adjacent to them. Cytoplasm is reduced to the sides. It is thin film is present at the side. It provides unrestricted pathway for the movement of food from source to the sink. Generally, sources are leaves. So food is transported to sieve tube elements, and then an unrestricted pathway is provided by these sieve tube elements. The nutritional requirement, since the cell is living, it is fulfilled. The nutritional requirement of sieve tube elements are fulfilled with companion cell, which is present adjacent to sieve tube elements. If you see the structure of companion cells have lot of organelles, nucleus, and these are present in adjacent to sieve tube elements, and they can transport food via plasmodesmatal connection to the sieve tube elements. They can fulfill the nutritional requirements, energy requirements of sieve tube elements. Next are phloem parenchyma. These are also living cells, and their main function is storage of food. When leaves synthesize food, maybe all of the food is not utilized or required by the plant at that particular time. So some of the food is stored in phloem parenchyma and can be transported to sieve tube elements in case sunlight is not available and food requirement is there and leaves cannot prepare that food. Then the stored food is transported to sieve tube elements, right? So last is phloem fibers. These are dead cells and basically provide strength to phloem tissue, not to the plant, to the phloem tissue. So these are the four components of phloem: sieve tube element, companion cell, phloem parenchyma, and phloem fibers. Together they form phloem tissue, and phloem tissue are responsible for the transport of food from leaves to all parts of the plant. Ideally, we should write from source to sink. It is a bidirectional pathway. It is a bidirectional transport. Source could mostly they are leaves, but sometimes when plant is not able to make the food from the storage organ, and it could be roots, food is also transported from roots to the aerial parts. So we say food is transported from source to the sink, wherever food is available, to parts of the plant where food is required. Where food is required, we call that part at that particular time as sink. So these are the two complex permanent tissues of the plant. Now, let's move to the third kind of tissues. If we divide plant tissues, if we categorize plant tissues on the basis of structure and function, we can categorize plant tissue into three types: meristematic, permanent, and protective tissues. So, meristematic and permanent tissues we have already discussed. Now we'll move to the protective tissues. As the name suggests, their main function is to provide protection to the plant. So let's study first type of protective tissue, that is epidermis. Epidermis is present over stem, leaves, roots, all over the plant body, entire surface of the plant. But root epidermis differs from stem and leaves. Epidermis over root, stem, leaves differ little bit, but there are many common features. So let's see those common features. Epidermis is basically single layer of flat cells, continuous layer with no intercellular spaces in between. These are flat cells. They form a continuous single layer with no intercellular spaces. One more feature of epidermal cell is that cell wall that is present on the outer side and on the sides. It is generally thicker on the sides and on the outer wall than the inner one. So as to protect the plant from the external environment. And for easy exchange of materials between the cells on the inner side, the inner cell wall is thinner compared to the outer. This epidermal cell layer, this epidermis, protects the plant from the water loss, from infections, from mechanical injury. So this is the main function of epidermis. It protects the plant from the excessive water loss, from pathogenic infection, and from the injury. Right? So also aerial parts of the, if we talk about the epidermis on the leaf, the aerial parts of the plant, wherever epidermis is there, we generally see a layer of cuticle. Waxy layer is secreted by epidermal cells. We call it cuticle, and it reduces the water loss. So this yellow layer is basically cuticle. This waxy layer prevents the water loss, excessive water loss from the plant. One more structure that is present in leaf epidermis is stomata, tiny pores. This is epidermis, but epidermal cells are interrupted by pores. If we see the epidermis leaves from above, we see something like this. These are modified epidermal cell. Epidermal cells become little bigger around these pores, and we call these epidermal cells as subsidiary cells. The pores are guarded by cells, kidney-shaped cells. We call them guard cells. And this pore, it is called stomatal pore. These three structures, guard cells, subsidiary cells, which are subsidiary cells are basically epidermal cells around the guard cells, which become little bigger than normal epidermal cells. So these three structure, we call them as stomatal pore complex. SPC, stomatal pore complex. So what is the function of stomatal pore? What are the function of stomata? These pores are present in leaf epidermis for gaseous exchange. Generally, plants have more of oxygen inside. So O2 generally moves out of the stomatal pore and CO2 moves in. So for the gaseous exchange, stomata are present. Second function that stomata perform is transpiration. Transpiration is basically loss of water vapor from the plant through the pore to the atmosphere. So the first function is gaseous exchange that will allow plant to do photosynthesis. Since CO2 is needed for photosynthesis and CO2 moves via these pore into the plant. So first function is gaseous exchange. Second is transpiration, which is needed for cooling of the plant, which is required for the cooling of the plant parts, and also it is needed for the ascent of sap, ascent of water and mineral from roots to the leaves. So both of these processes requires the presence of stomata on the leaf epidermis. So far we have seen epidermis in case of normal terrestrial plants. Now let's see epidermis in case of desert plants. Desert plants are those plants which face shortage of water. So they need to save more water compared to the normal land plants, since water availability is not there. In desert plant, if we cut the section of the root, we see something like this. They have multi-layered epidermis. Also, the epidermal cell secrete, they secrete over their surface. So they are covered with thick waxy layer of cutin, we call it cuticle, to prevent water loss. So multi-layered epidermis is present in case of desert plants. Now let's see the case of roots. If we cut the section of root tip, we see epidermis looks something like this. These are epidermal cells. This is epidermal layer. And some of the cells show projection. Plasma membrane protrude out. Cell wall and plasma membrane protrude out to form root hair. So these are the special epidermal cells which have root hairs. These root hair increases the absorptive area, surface area for water absorption. So the area for water and mineral absorption increases because of the formation of this root hair from the epidermal cells. So now let's move to the second protective tissue, which is cork. Cork is made up of dead cells. These are formed in the older stems and branches. This figure here shows, this photograph here shows an older stem, and we can see the presence of cork over its surface. So cork is generally formed in the older stems and branches. Cork cells are dead, compact cells with no intercellular spaces. So these are dead, compact cells that lack intercellular spaces. So where are they formed? If we cut the cross section of the stem, this is the epidermis. And in the cortical region, this is the cortical region, cortical layer, some of the cells in the older stem become meristematic. We call it cork cambium. And it cuts off cell on the outer side. It cuts off dead cells, which are compact, dead, and water and gas proof. They do not allow water or gas to pass through because these cells have thickenings of suberin over them. They have suberin thickenings, so that makes them waterproof and gas proof. So cork cambium keeps on cutting off multiple layers of cork, as a result of which, because of the pressure, epidermis in the older stems gets ruptured. So this is the ruptured epidermis because of the multiple layers of the cork cells. Cork cells provide protection to the older plant parts. Since the cells are waterproof, gas proof, it does not allow stem to get affected by pathogenic infection because water and gases are not going in. So it provides stronger protection to the plant part as compared to epidermis. So this is the second type of protective tissue in plants. So that's it for plant tissues. In the next session, we are going to study animal tissues.